Ladies and gentlemen, welcome back to the Global Young Scientist Summit. My name is Preston Ching from the National Research Foundation Singapore, and I'll be your MC for the rest of today. I hope that you have settled down after some engaging conversations over lunch and are ready for our next plenary. For those of you who are seated at the back, feel free to come on down and fill up the seats in the front as well. So without further ado, let's welcome Prof Brian Kobilka to talk about structural insights into G protein coupled receptor activation. The Q&A session will be moderated by Prof Van Antoine. Prof Kobilka, please. Thank you.
Thank you. So I want to thank the organizers for inviting me. So we're going to go from something very big, the universe, to something very small, and talk about this family of proteins that are very important in regulating virtually all functions of the human body. And these are G protein coupled receptors. So what are G protein coupled receptors? They mediate the majority of our body's responses to hormones and neurotransmitters. And shown here is the basic paradigm of how these work. So this is a receptor in a membrane. A hormone or neurotransmitter will bind to the receptor, change the shape of the receptor so that it is now recognized by proteins called G proteins. And these include three components: an alpha, a beta, and a gamma subunit. When the activated receptor activates G proteins, the G protein will release GDP and bind GTP, dissociate into an alpha subunit and a beta gamma subunit. And these two proteins then regulate various enzymes or channels within the cell. And that regulation continues until GTP is hydrolyzed to GDP, the subunits reassemble, and the cycle can begin again. So what are hormones and neurotransmitters? Well, you probably know this anyway, but neurotransmitters are released from neurons. So neurons talk to each other through neurotransmitters. Nerves also talk to, for example, skeletal muscles and various other cells within the body. And examples of neurotransmitters that are activating GPCRs include noradrenaline, or dopamine, serotonin, glutamate, endogenous opiates, as well as heroin and fentanyl, and endogenous cannabinoids. So these are all examples of neurotransmitters that activate G protein coupled receptors. Hormones, even more. And hormones can act locally, where, for example, cells from the immune system release various hormones that modify the function of other cells in the immune system. And they can also be released into the bloodstream and then affect cells at a great distance. And examples of hormones include histamine, adenosine, prostaglandins, and angiotensin and glucagon. These are all examples of hormones that work through G protein coupled receptors. In addition to mediating hormones and neurotransmitter responses, they mediate the senses of sight, smell, and taste. There are over 800 members in this family. So it's the largest family of membrane proteins in the human genome. So here's the family tree. And the family tree can be divided into the major class. Before I go in, I'll note here, so this is what we call family A, and the majority of our 800 or so G protein coupled receptors are dedicated to detecting the sense of smell. So this is just a point of interest. Humans really don't rely on their sense of smell that much to survive. I can tell you from personal experience, I no longer have the sense of smell, and I'm still here. But other animals really rely on their sense of smell, and they often have more olfactory receptors. So, for example, mice and rats have over 1,000 olfactory receptors. And I think, surprisingly, elephants have over 1,000 olfactory receptors as well. So this is the largest subfamily called family A. It includes receptors for some hormones and neurotransmitters that you might recognize, such as adrenaline, muscarinic receptors for acetylcholine, dopamine, serotonin, opiates, cannabinoids, and the range of activators is really truly remarkable, from photons, for example, for vision, to protons. Some receptors are dedicated to detect changes in pH, to small molecules like adrenaline, to peptides, and even to relatively large proteins. So these are remarkably versatile membrane proteins. My lab also works on some what we call family B and family C, but I'm only really going to focus our efforts on trying to understand family A. Because they're so important in detecting hormones and neurotransmitters, they have become the single largest class of pharmaceutical targets. And for this reason, we've been very interested in understanding how these proteins work in molecular detail. We want to know what they look like in 3 dimensions, how they engage their signaling proteins. And ultimately, this information can be used by the pharmaceutical industry to develop safer, more effective, more selective drugs. So if you want to know how a protein works, you need to really know what it looks like in 3 dimensions. And that's been a goal of my research for over 30 years now. So you can start by learning something about proteins if you can clone the DNA that encodes them, or clone the messenger RNA that encodes them, and then you can determine at least the primary amino acid sequence of the protein. And in some cases, if you just know the primary amino acid sequence, you can ultimately determine the 3-dimensional structure. And in fact, there are now programs available where one can accurately predict many GPCRs without ever actually getting experimental data on their structure. So a brief history of the field related to understanding the structure started about in 1971 when Marty Rodbell proposed the existence of G proteins as being mediators of signaling by these class of receptors. It wasn't until 1977 that G proteins were actually identified biochemically and purified by Ross and Gilman. And Gilman and Rodbell would ultimately get a Nobel Prize for their work on G proteins. The first purification of a GPCR for a hormone or neurotransmitter wasn't until 1984, and this ultimately led to the cloning of the gene for the beta-2 receptor. So this is a sort of a remarkable piece of biochemistry that I'd like to show. This is, again, the purification of the beta-2 adrenergic receptor. The beta-2 receptor is abundant in lungs. It's an important target for asthma. So this postdoc in the lab of Robert Lefkowitz, his name's Jeff Benovic, did this really Herculean purification, 33,000-fold purification of beta-2 receptor from half a kilogram of hamster lungs. Approximately 1,000 hamsters were required to achieve this. And from this, he was able to get 50 micrograms of protein, a very small amount. And then he chewed that protein up with a protease, isolated the peptides. And from the peptide sequence, we could guess what the DNA sequence would be that encoded that peptide. And that DNA sequence was used to clone the gene for the beta-2 adrenergic receptor. So now we have an uninterrupted coding sequence for the receptor. We can translate it and get the amino acid sequence. And then we could detect from the amino acid sequence that it contained 7 hydrophobic sequences suggesting 7 membrane spanning domains. And this was our first clue as to what these receptors look like. It's basically the secondary structure. We don't have the tertiary structure. But from this, we could begin to do a number of experiments to try to figure out where the binding sites were and where the G-protein interaction interface was. And I'm going to talk more about that in the next few slides. But so after the cloning, the next milestone in the field came when the Gilman group obtained the structure of a heterotrimeric G protein. It wasn't until 2003, after the human genome was sequenced, that we realized that the family was so large, with 800 members. And then in 2007, we were finally able to get the first structure of G protein coupled receptor in the inactive state. This, again, was the beta-2 adrenergic receptor. Several years later, we were able to get a structure of the beta-2 receptor in the active state. And then at the end of 2011, we were finally able to get a structure of a receptor caught in the act of activating as G protein. So why did it take so long to go from the cloning when we actually knew what the protein sequence was? We had the DNA, we could start making protein. Why did it take so long to get from having the clone to understanding what its structure is? And so I'm going to give you a little idea about why these proteins were somewhat difficult experimental targets, and it's related to how versatile they are as signaling molecules. So if you want to know the structure of a protein, particularly back more than 10 years ago, you needed to be able to grow a crystal of the protein. So the protein has to be very pure. A crystal is an ordered array of the molecules. Once you have a well-diffracting crystal, you bring it to a synchrotron, shine X-ray beam through the crystal. You can, from the pattern of the diffracted X-ray beam, you can calculate what its three-dimensional structure is. So this requires first that you be able to make enough protein. And again, having the clone allowed us to make some modifications to the protein so that it was much easier to purify the protein than it was to purify it from native sources. As an example, we put an epitope for an antibody that recognizes the receptor. You can put another epitope at the end of the protein, again, use another type of chromatography. And with these tools, we were able to make enough protein to begin crystallography studies. So we express protein in insect cells. The protein is embedded in membranes, and so it has to be extracted from the membranes with detergents, which are membrane surrogates, and then going through several purification steps. And ultimately, you have pure protein that's fully functional, and it can be used for a number of biochemical studies, biophysical studies, and crystallography. So in spite of having purified protein for about 10 years, it took from the first time we could make enough protein till we got our initial crystals was about 8 or 9 years. And for those of you who have done crystallography, will realize these aren't very nice looking crystals, but they're the best we had. We brought them to a synchrotron, and we had no diffraction spots. And one of the problems that we recognized is that these crystals are very small, and these synchrotron beams are quite large. And we felt that if we could use a smaller beam, that we might actually have better signal to noise. And we were very fortunate that in 2005, Gebhard Schertler was working with colleagues in Grenoble to develop the first microfocus beam. And he brought us to Grenoble, and we had our first evidence of diffraction. So this is, again, one of our crystals. A conventional X-ray beam would be roughly this size, so you have a lot of noise where you and relatively small amount of signal. However, the new microfocus beams were 5 microns, and from that first diffraction pattern, we knew we had protein crystals. But the further out these spots go, the better view you have of the protein. And these were very poorly diffracting crystals. So in spite of having now some diffraction spots, we still weren't able to determine the structure of the protein. So why was that? So at the time, most of the behavior of receptors could be explained by this simple two-state model, where a receptor was either in an inactive state or an active state that could then engage the G protein. And under this condition, one would expect relatively stable states, either in the inactive or the active state. However, studies that were being done using other methods, biophysical methods, revealed that this two-state model was just an oversimplification, and that these proteins were actually highly dynamic, that the parts of the protein that recognized the G protein were in an ensemble of conformations, and that this heterogeneity made it difficult to actually crystallize a protein. So receptors, rather than looking like solid objects, were, as shown here by this molecular dynamic simulation, highly dynamic, and as a result, very challenging to crystallize. So, and we also learned from these studies that there were two transmembranes, particularly transmembranes 5 and 6, that were especially dynamic. Now, why does that make a difference? Well, I'm going to simplify the crystallography problem by using an example of a two-dimensional crystal. So if we have a conformationally uniform protein, they behave like these regular hexagons. And you can pack the hexagons nicely into an array. However, if you have a more dynamic protein, even though you have pure protein, each of these protein molecules might look slightly different from its neighbor. And as a result, you have a poor crystal quality. So we had to try to overcome these problems. One was the problem of protein dynamics. And the other problem was that the receptor was embedded either in a lipid bilayer or a surrogate of the lipid bilayer. And there weren't many options for a protein crystal to grow, because proteins typically will crystallize where the hydrophilic parts of the protein are connecting with each other. And so we took two different approaches. One was to stabilize the protein in an inactive state with an antibody, or to do so with protein engineering. And it turns out that both of these worked at about the same time. We now had protein crystals that looked more like protein crystals, and they diffracted much better. So now, instead of 20 angstroms, the spots go out to 3 angstrom resolution. So this was the receptor bound to an antibody, and this was the receptor engineered to have a small protein called T4 lysozyme, replacing the loop between TM5 and TM6. And this was also done in a special kind of lipid. The lipid itself is sort of an ordered array of lipids called a cubic phase. And as a result, we were able to get even better quality crystals. So having an antibody or having this protein T4 lysozyme allowed, stabilized the two most dynamic parts of the protein and allowed for crystal packing to occur. So both of these structures were available to us in 2007. Now the next challenge, we have the inactive state. The next challenge was to get the active state. And this was particularly difficult because, again, proteins are quite dynamic. However, we were able to get a crystal structure of a receptor bound to a covalent agonist. So it's definitely in the inactive state. Or the agonist is a covalent agonist. However, when we got the crystals, it was in the inactive state. So again, that goes back to our model, in that our model sort of predicts that even though in the presence of an agonist, some of the receptor will exist in an active state, the majority of the receptor is still in the inactive state. And you actually need some sort of stabilizing protein, which could be an antibody or a G protein, to stabilize the active state for crystallography. So we were trying at the time to get a receptor G protein complex, which was unsuccessful. However, we then started using a relatively new antibody technology, which were camelid antibodies or single domain antibodies called nanobodies. So these camelid antibodies, you can see, have single heavy chain compared to antibodies that we generate, which have heavy and light chain. And as a result, the variable component is a small oval-shaped protein that fits well into protein crevices and is useful for stabilizing them. So having a nanobody that recognized the active state of the receptor allowed us to get the first structure of an agonist-bound active state of the receptor. However, our major goal was to get this structure, the receptor G protein complex. And this was even a more challenging project because G proteins are somewhat challenging to make, and receptors are also, as I mentioned, challenging. We then had to get them to assemble and remain stable. And we were able to ultimately do this through a collaboration with Roger Sunahara. And for those of you who aren't familiar with the biochemistry, this is just to show that we had absolutely pure protein. The complex was very stable. It gave us this single peak, suggesting that all the receptor was complex to G protein. So this looked very encouraging. However, we were unable to grow crystals of this. And one of the reasons we ultimately found out is that now the receptor was stabilized in a single active conformation. However, when the G protein loses its GDP, it becomes highly flexible and dynamic. So we had another problem with protein dynamics. And this is just a list of the contributions that had to be made in order to get this to work from, as you can see, scientists from around the world. This included our experts in G protein purification, Roger Sunahara. We had to identify highly efficacious and high affinity agonists, which several pharmaceutical companies sent us their pool of beta receptor drugs. We had a new detergent. So again, detergents are used to replace the membrane. And this detergent was made by a detergent chemist, Bill Suk-che. Wasn't available commercially. He sent it to us, and this was really critical for stabilizing the complex. We had some protein engineering. We had a nanobody that helped stabilize this dynamic component. And then a lot of our work was really guided by negative stain electron microscopy. And then finally, we had access to a new detergent, a new lipid for making cubic phase, again, not available commercially by Martin Caffrey. So after all of these contributions, we were finally able to make the beta-2 receptor G protein complex crystallize. So what did we learn now that we have a receptor in an inactive state and an active state? And first of all, the binding pocket for this receptor is formed by the transmembranes and the extracellular side of the receptor. The binding pocket is rather small, and you can see that the major interaction between the receptor and the G protein was at the very C-terminal end of the G protein, engaged into the core of the receptor, which is opened up by the outward movement of TM5 and 6. So in the next few slides, I'm going to be going back and forth, going from active to inactive. And what's sort of remarkable is that the changes that occur in the binding pocket are relatively small, only about a couple of angstroms change. This propagates to a much larger change in the cytoplasmic side. So transmembrane 6 moves out about 14 angstroms. And then these translate into much larger changes in the G protein upon its activation. And you can see the G protein engaging into this core. So now we have a beta-2 receptor coupled to a specific G protein. As I mentioned, there are 800 other GPCRs. There are also a relatively large number of G proteins. And we wanted to know whether what we were learning about the beta-2 receptor coupled to the G protein GS worked with other receptors. So we began forming complexes of other family A members with different G proteins. And we were able to form nice complexes. None of them would crystallize. So this was around 2017. And up until then, we had been relying entirely on X-ray crystallography. And as I mentioned, you have to be able to grow crystals. Fortunately, around this time, cryo-electron microscopy had advanced considerably with new detectors and also algorithms for collecting and accounting for motion that occurs and new refinement methods. And this is a really important advance because you don't need crystals to get structures by cryo-EM. You simply take a large number of two-dimensional projections, and from these two-dimensional projections, you can calculate a three-dimensional structure. So having access to now cryo-EM, we were able to obtain a number of other receptor G protein complexes, and it turns out that they're all pretty much the same. They have the same conformational changes in the receptor, very similar, if not the same conformational changes in the G protein. The only difference being family C, which are really quite different, and I don't have time to talk about that. So what have we learned? First of all, the nucleotide-free complexes are similar for family A GPCRs that couple to different G proteins. Structural changes in the G proteins upon activation are very similar. The alpha helical domain, this part of G protein, is highly flexible for all G proteins. And one thing that was quite disappointing is that the receptor G protein complexes that we obtained did not fully explain why a particular receptor will prefer a particular G protein. And we still don't really understand this. And what we do believe, though, is specificity is probably determined by some sort of intermediate state. And we have evidence that between the binding of an agonist and the coupling of a G protein, there are probably at least one, if not more, transient intermediate states, which are probably critical for determination of the structure. So what use are these structures? Well, structures have been used to identify new drugs using in silico and computer-based docking programs. And I'm just going to briefly contrast the old method. So the conventional way of screening for drugs for G protein coupled receptors involve testing each compound. So the compound libraries typically are around 1 million compounds. And all of the compounds have to be tested on cells expressing receptor individually. So these libraries have what we call relatively limited scaffold diversity, so that even though there are 1 million compounds, many of these compounds look like other compounds in the library. Experimental testing is slow and expensive. And so now having access to high-resolution three-dimensional structures, it's possible to do screening entirely by computational methods. So using docking algorithms together with a crystal structure, the virtual libraries are now roughly close to 1 billion compounds. They have high scaffold diversity, and it's far less expensive than screens. So these computational programs essentially take each of the compound in the library and test it in a large number of different orientations, and if it fits, it gets a high score. If it doesn't, it gets rejected. So this is more or less the state of drug discovery for the field, and it's become much more useful for both drug companies and academic labs now to identify novel ligands for GPCRs. So I'm going to end here, and I just want to acknowledge a large number of collaborators who have really helped with the project throughout the years, and people from my lab. And I thank you for your attention.
thank you, Prof Kobilka, for very interesting talk. A lot of work, and I can see that you have overcome many challenges along the way to achieve the final goal. So each time you analyze a situation and you find a solution to the problem. But I guess there are many failure attempts along the way, right?
Yes.
Can you share with us how is there any time that the team is lost, that they couldn't find a solution?
Well, at least for this work, ultimately we did find a solution, but as you can see, it involved a number of collaborations outside of our lab, and often going to scientific meetings where you hear people from different disciplines present, often giving you ideas on how to, how to solve your problem using approaches that generally haven't been applied to these receptors.
I see, yeah. So the audience open for the question. Any question in the audience? Yes, please, can you go to the microphone? Can you go to the microphone?
Good morning, everyone. Myself, Ashish Srivastav from Shivnadar University, India. So I have a question regarding GPCR. How we can target a GPCR which is mutant and we can regain its activity?
I'm sorry, could you repeat that?
Yeah, a mutant GPCR protein for class.
A mutant GPCR protein?
Yeah, GPCR. Class C GPCR.
Yeah.
So it is a loss of function mutation. How we can regain its activity by a small molecule? Is it possible?
So there are some examples where that's been done. There are receptors that are, they're called vasopressin receptors. They're important for controlling fluid balance in the body. And there are some mutations that prevent proper folding. And they don't get to the plasma membrane, so they don't work. Nevertheless, they'll bind small molecules. And so by giving a patient a small molecule that binds to the receptor, it will stabilize it enough so that it makes it to the plasma membrane. So that's the only example I know, and it's a mutant that affects mainly folding and not binding. I would say if the mutation completely prevents binding, then there's probably nothing you can do in terms of developing drugs to solve that problem.
Okay. And is there any impact on the intercellular disorder region when we don't take in the MD simulation study?
Right. Oh, yeah. So that's an interesting question. So there are basically 3 disordered regions: the amino-terminus, the loop between transmembrane 5 and 6, and the C-terminus. And we have studied the beta-2 receptor C-terminus, and it turns out that it's a regulator of G protein access. So the C-terminus actually, while it's disordered, it folds into the core, cytoplasmic core of the protein, and essentially prevents G protein from getting access. So it reduces signaling.
Okay. Okay. Thank you very much.
You can think more about the question. I have a question coming in. Display. Okay, let me read. Do you think it will be ever feasible to image this protein receptor dynamically over time at a couple?
Actually, watch it. So I think it's probably possible to do that by cryo-electron microscopy. And my colleague, Yorgos Kiniotis, is doing something like that. He's actually watching it dissociate. So he adds G protein and freezes a grid and adds GTP to cause the protein to dissociate, and then freeze the grid. And what you could see from one sample of frozen receptor are several steps in the process of dissociation. It should be possible to do that in the other direction, to just add receptor and G protein, and if you time it right, to be able to actually watch assembly of the receptor G protein complex. it just will take a lot of time, a lot of microscope time, a lot of computational time, but I think it's possible. It may not be possible to capture really transient states that are important, but we'll see.
I have another question. So since GPCR are the largest class of pharmaceutical targets, are there big challenges in drug design in off-target toxicity? How does one overcome them? So the question is on, since the receptor is a major target, so what the problem of off-target? Because you also mentioned about the specificity.
Yeah. So that's a really good, the off-target effects are really a large problem. So for example, there are 9 different receptors for adrenaline. Each one does something different. Their binding pockets are highly, if not identical, very similar. So when you try to develop a drug for, say, a beta-2 adrenergic receptor, it will also often hit a beta-1 adrenergic receptor or an alpha adrenergic receptor. And that's true for serotonin receptors, dopamine receptors. And in fact, serotonin and dopamine receptors are very similar to each other. So many times when you're trying to develop a drug for a serotonin receptor, you'll hit a dopamine receptor. And it is useful to have structures, particularly structures bound. So let's say you have a drug that initially is non-selective. If you can get a structure of the receptor bound to that drug, you can perhaps learn how to change the drug so it's more selective by extending out of the highly conserved pocket into parts of the receptor that are more diverse. Another way of getting more selectivity, fewer off-target effects, are to develop allosteric drugs. So allosteric drugs bind outside of the native hormone binding pocket. Often they can bind almost anywhere in the protein and either enhance or inhibit responses to native hormones. So the advantage there is that just the fact that they're modulating the response to native hormones might make them more safer. And the fact that they're binding outside of the highly conserved pocket means they're likely to be more selective. So there's a greater interest in developing allosteric drugs for these receptors.
But then should drug target the active state or inactive state, or in between that we don't see?
You can do both. Yeah, you can actually target, both. One of the approaches that we've been using are DNA-encoded chemical libraries. You essentially have libraries of billions of compounds. Each one has a DNA barcode, and you look for compounds that stick to receptors in specific states. And it's been possible to find positive and negative modulators using this approach.
Any question in the audience? Yes, please.
Good afternoon. I'm Sin from Thailand. My question is, do you think your artificial intelligence will be a paradigm shift of drug discovery?
Could you repeat that?
Do you think artificial intelligence will be a paradigm shift of the drug discovery? Like the AlphaFold will help you to do drug discovery or accelerate your molecular screening?
So I think AlphaFold is a significant advance, and I think it will allow determination of structures without getting either by cryo-electron microscopy or crystallography that are good enough for in silico screening. I think it's still probably helpful to have a structure if you've identified a drug and you want to make it better, to actually have a structure with the receptor and bound to the drug. I don't believe AlphaFold is as good at predicting the location or orientation of the molecule. One thing that AlphaFold won't do is predict allosteric binding sites yet.
Thank you very much.
Any other question? Maybe we can one question here.
Okay.
Are there any?
This one.
Okay, please go ahead.
Yeah, thank you. Good morning to everyone. So I'm James Paul, representing Philippines in Japan. So basically, my question is out of the topic because I know that most of the questions are being asked is technical about the. So I saw that your presentation or your research already experienced many challenges about doing research about the GPCR. So as a young scientist here, we are young scientists here. So how can you overcome these challenges? Can you tell us your strategies and your motivation so that we are all motivated here as a young scientist? Thank you.
I think in most cases where we've overcome challenges, it's sort of stepping outside of the field itself. And by attending meetings where there's a sort of broad range of science, you often can think of, well, maybe that approach will work for me in this particular problem where it's never been applied before. But unfortunately, it's persistence, a lot of luck, and in my case, having really talented postdocs and graduate students and colleagues to help out.
Yeah, thank you very much. That's well noted.
So I noticed that along the way when you, so first you clone the DNA when the DNA sequence, human genome was not available, right? So if, and then the technology evolved and it's available. And then back then EM was not available and now it's available. A lot of things can be solved. So,
Yeah.
so I guess there's still a lot of problems that we can solve faster these days.
Yes, yeah, I mean, the development of cryo-EM has been just a major advance in the field. Now, it's not that challenging to get structures anymore. I should say there's also a lot of luck that happened. For example, at the time we were trying to clone the receptor, it was typical that you would do a cDNA library, and cDNAs are copies of messenger RNA. We were unable to get a clone. There were just the abundance of cDNAs were too rare. So we finally decided to go for a genomic clone. And typically, most proteins in the human genome are composed of exons and introns. So at best, we'd expect to maybe get an exon that would help us. But it turns out the beta receptor is intronless. So the entire coding sequence was present in the DNA and the genomic clone. So that's very lucky.
I see. Yeah. So you need a lot of luck in research. Okay, maybe we can go to some of the questions here. Are there any nutritional compound that bind to GPCR that can be seen in therapeutic in future?
Well, I know there are receptors that bind to metabolites of food, and actually metabolites of the gut microbiome as well. There are olfactory receptors that are not only in the nose but in the gut, and they can detect at least metabolites. It turns out that a lot of your taste is mediated by G protein coupled receptors. So sweet, sour compounds, umami are all mediated by G protein coupled receptors.
So then we talk about COVID, people lose taste. Does that mean those receptors are damaged?
Yeah, I think most people who have COVID probably experience more olfactory.
I see.
Yeah, but of course that affects your taste, sense of taste as well.
I see. We have one last question. If anyone have question, yes, please.
Hi, good afternoon, everyone. So I was just wondering, see, if the family of G proteins are really, so if within a family, the structure is really conserved, but you have different specificities of the G protein itself, could it be that the G protein specificity actually depends not on the structure, but on something else? Is that a possibility?
Since we can actually, we can determine specificity using purified systems, we're excluding other things. I think I would have to say it's largely based on structure. But things that do influence structure can influence the selectivity. For example, the composition of the membrane. What lipids are in the membrane might influence the way it interacts. For example, the beta-2 receptor will couple primarily to a protein called GYSS. It will also couple to GI, but only in a certain membrane environment. But again, that membrane environment is probably determining or modifying the structure of the protein.
Thanks.
Since time is up, so let's thank Prof Kobilka for the amazing talk.